Good morning, Dr. Sumpei.
Sorry, sorry for late.
Yeah, okay, no problem.
So, can I start?
Can we start now? Yeah, okay, thank you so much. So I will start the UGC in the second term. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. So this is the second session of the UGC with Dr. Sumpei Hatta from University of Malaysia Terengganu. Today we will get the explanation about the trial associated from the Dr. Sumpei Hatta. Before we start our UGC today, we have to take a photo sessions. So please open your camera first to take the photo session. Okay, thank you for the photo session. Hmm. Now we have Dr. Sumpe in our Zoom meeting today's. Um, Unfortunately today, Pak Anto cannot attend the Zoom meeting today's. So this is me for as moderator, and also we have our narasumber, Dr. Sumpe. Yeah. For the UCC today, Dr. Sumpe, please welcome.
Okay. Good morning, everyone. Today, I want to share about the trust criteria. Last week, I shared about the microbiome. So today is the sharing the about the trust criteria. Okay. Everyone can see the what is trust criteria? The slide. Okay. So maybe the all of you don't know what is the transcriptase. Transcriptase is a group of marine proteins and which are close to the fungi. So normally the transcriptase is the fungi-like proteins. And this transcriptase is a single cell organism and found in the diverse environments such as the ocean and brackish water. And normally the easy to find is the mangrove or the mangrove reef because of they are the one of the decomposer. So this is the morphology of the transcriptase. Then this one, we can easily observe the transcriptase using a microscope, even though that we can observe that their colony using the microscope, not like the bacteria. So their characteristic, their size is 4 to 20 micrometer size, and they produce the ectoplasmic net, this EN, and the granular cytoplasmic and the globose and the subglobose shape. And they have a lot of the lipid content, and they have the biflagellate zoospores and the non-cellulosic cell wall. And they also produce not only the lipid, they produce a squalene and the carotenoid, as well as the hydrolytic enzyme. So what kind of lipid they can produce is the PUFA, MUFA, SFA. PUFA stand for the polyunsaturated fatty acid. And the MUFA is a monounsaturated fatty acid, and the SFA is a saturated fatty acid. I'm not sure that everyone know what the difference. One is saturated is like the no all the connection is saturated, and the mono is the only one bind is the double bind. Then polyunsaturated is the many of the two or three of the unbind one, double bind one, and PFA compose the DHA, EPA, arachidonic acid, and alpha linoleic acid. And MUFA is a representative of the oleic acid, and the SFA represent the palmitic acid and the stearic acid. So, the definition and the taxonomy of the trouscaitrid. Definition of the trouscaitrid is heterotrophic marine Stramenopiles, part of the family class Labyrinthromycetes. They are microscopic eukaryotic organisms found worldwide, and their taxonomy is initially categorized with the fungi due to their heterotrophic nature, and but they are now classified as the Stramenopiles, which include the diatom and the brown algae. So some the researcher say the trouscaitrid is the one of the microalgae. But as you all know, microalgae have the chloroplast, and they can do the photosynthesis. But the trouscaitrid, they don't have the chloroplast, and they cannot do the photosynthesis. So the trouscaitrid is not microalgae. That's why I say that trouscaitrid is a fungi-like protist. So their evolutionary history is they share a common ancestor. The common ancestor with the diatom. This one of my side is here, labyrinthrite or the trouscaitrid. Same with the ancestor with the diatom and the brown algae. And demonstrated their complex evolutionary history and unique adaptation for the marine life. So that means the very difficult to find the trouscaitrid from the fresh water. Normally, we can easy to find the marine water and brackish water. Okay, and this is a taxonomy. So Chromista, Harosa, and the Storamenopiles. Then class Labinichromycetes. Then class to Chaetoriales. Then Trouscaitrid family. Then they have the Oblongichaetoriaceae also considered Trouscaitrid, but nowadays they have the standalone the family. Previously, they are under the Trouscaitoriaceae. And under that family, composed the Trouscaitrim, Japonochaitrim, Schizochaitrim, Auranchaitrim, Hondaia, Archenia, Schizoideo, Chaitrim, Parietichaitrim, and Monorizochaitrim, and Botryochaitrim. So they are the very different character. And this Apranochaitrim also previously considered in the trouscaitrid, but nowadays the changes. But maybe the change back to this is the 2019, but still the many researchers, trouscaitrid ecologists, discuss the really this taxonomy is correct or not, because they are very diverse the characteristic and the DNA also, and the life cycle also very unique. Life cycle I share the next slide. So based on this kind of one, still Triskaideraceae or the laminaturales is the many changes. So this is the reproduction and the life cycle of the Triskaideraceae. So normally vegetative the reproduction and to produce a zoospore, this is a normally. So the most of the Triskaideraceae have this life cycle. But some cross criteria have the amoeboid loop or the binary division, or some might have the sexual reproduction also. So that's why their life cycle is unclear. That's why I just said the taxonomy also not confirmed yet. Okay. Then habitat and the distribution is mainly the marine environment. Triskaiderae are primarily found in marine environment, partially in the coastal water or the estuary and the intertidal zone, and they thrive in the nutritional and rich area with a high level of the organic matter because of they are the one of the decomposer. That's why previously the researcher or scientist thought Triskaiderae is the fungi because the fungi is one of the main decomposer of the environment. And sediment and the planktonics also, that they are commonly associated with the sediment or the debris like the leaf. As I showed this photo picture. This is the foreign leaf inside the marine mangrove leaf and sediment and can also be found in the planktonic community, which they play a role in the nutrient cycle and the food web. So planktonic community means just the free living on inside the water. But of course, they are the different type of the Triskaiderae with the marine water and the brackish water because the example, the Aplanocytorium, they are the zooplankton or the plankton eater. So they can easily to isolate from the marine water. And when we isolate, we need to put plankton as the prey. Then global distribution, the exhibit wide geographic distribution, so the worldwide. So Malaysia and Indonesia and Japan, even though the Antarctic area also found the Triskaiderae. So they have a lot of the ecological roles. At first, a primary producer. They are heterotrophic organisms that play a vital role in marine ecosystem by consuming the organic matter such as algae and bacteria and converting it into the biomass like the pupa or squalene or the many the protein source and nutrient cycle. They also contribute to nutrient cycle by releasing the essential nutrient like the nitrogen and the phosphorus back into the environment. So which supporting the growth of the other marine organisms. And food web dynamics also same with the as a serve as a food source for various marine organisms, including zooplankton, fish, and the shellfish, and contributing to the complexity and the stability of marine food web. So some marine, mainly the marine fish, they cannot produce the pupa. Marine fish sample cannot produce the pupa, but they have a lot of the pupa. This one might be because of they intake the microalgae and the triskaiderae. So Triskaiderae is one of the very important source for the fish and the shellfish. In the microbial interaction, the Triskaiderae participate in complex microbial interaction, forming a symbiotic relationship with other organisms and influencing the composition and the function of the microbial community. This one is still unclear, So that nowadays the scientists also investigate how symbiotic relationship with which microorganisms. Other than the ecological roles, they have the biotechnological potential. Maybe the transcriptase is the most part is this part. So transcriptase are the gaining the recognition of as the sustainable source of the high value products, including a lipid, carotenoid, enzyme, and the biocatalytic with application in food, pharmaceutical, and nutraceutical and the biofuels. So because of high lipid content, transcriptase are known for their ability to accumulate a high level of lipid ranging from the 20 to the 60% of their dry weight, and that their lipid fatty acid profile is very diverse in the variable PUFA, including DHA and the EPA. And their nutritional value is the high PUFA content make them a valuable source of the nutrient for human and animal consumption. So let's see the bio, and other than that, biofuel potential transcriptase are being explained for their potential to produce biofuel, especially the biodiesel, because of their high lipid content, like the saturated fatty acid or the monounsaturated fatty acid here, and the carotenoid and the pigment production. They produce the wide range of the carotenoid and the pigment. So this contribute to the vibrant color and the antioxidant properties. And the production of these product pigment hold the potential of the various industry, including food, coloring, cosmetic, pharmaceutical. And their biomass themselves also very important. The transcriptase are sustainable and a very valuable source of the PUFA for fish feed, supporting the fish health growth and the development in the aquaculture, and they can be integrated into the mariculture system to contribute to a circular economy by converting waste into the valuable products and promoting the sustainability. And other than that, also the pharmaceutical use, like the anti-inflammatory agent, like the DHA and the EPA, known as the anti-inflammatory property, and the cholesterol reduction also, and immune system support also. That's why nowadays the many researchers in the industry the focus on the transcriptase as the future bioresources. Other than that, enzyme and biocatalysis also important for the as the transcriptase. So because of that, they produce hydrolytic enzyme. So they produce the variety of the hydrolytic enzyme like the lipase, protease, kitchenase, and the cellulase also. These kind of the enzyme play important role in their nutrient acquisition and degradation of organic matter. And the biocatalysis for the industrial application. So this enzyme can be harnessed for industrial application such as the biofuel production or the bioremediation and the production of the variable chemicals to use their enzyme. Thus, that's why the production of the enzyme present the opportunity for developing a novel biocatalytic process in various industries. So this is just now I showed the bioremediation here. So this is the biodegradation and the bioremediation for the as the using the transcriptase. So for the bioremediation, transcriptase ability to degrade the pollutant makes them promising candidate for bioremediation application, and transcriptase might help to remove pollutant from the contaminated water and the soil. And the biodegradation also same. The transcripts play a role in the breakdown of the organic matter in marine environment. So they can degrade a variety of the compound, including the carbohydrate, lipid, and the protein, because they have the variety of the enzyme. So now I move on to the how to isolate or cultivate or the fermentation. So at first, the strain selection. This is very important for the further step of the industry because if we don't choose properly, then the final products cannot get the desired products and the high quality. And after that, culture media optimization. Optimize culture media composition, including carbon source, nitrogen source, and the trace element, is essential for optimal growth and the product formation. Later, I shared my own result about the culture, how culture media influence their lipid compound. And strain selection also I will share with you. And fermentation process design is choosing the appropriate fermentation system like the batch, fed batch, or continuous, depend on the specific strain and the product's requirement. So if the SSFs are solid state fermentation, maybe just batch or fed batch. But if the submerged fermentation SMF is using the fermentation system because the continuous fermentation system mean that every time the transcript get the fresh environment and no toxic exhaust waste. And process control and the monitoring. Monitoring key parameters such as pH, temperature, and the DO is very important to maintain the transcriptory growth condition and the product quality. And other last stage is downstream the procedure. The efficient downstream processing technique are need to separate, purify, and concentrate and the desired product. So the design is very important also. If the contaminate, then we cannot get the proper product, final products. Next is a nutritional requirement. So as I just say, carbon source, nitrogen source, phosphorus, and the mineral very important to the transcriptory growth. And the lipid compound or the beneficial compound. So carbon source, why important? Because transcriptory are heterotrophic, relying Transcriptory is a heterophyte relying on organic compound for growth, and they utilize a variety of the carbon source, including sugar, lipid, and organic acid. So carbon source case is the glucose, fructose, or the other polysaccharide, or the glycerol is the considered carbon source. Then nitrogen source is the transcriptory require nitrogen for protein synthesis in the cellular process. And so nitrogen source can include nitrate, nitrite, and ammonium as the inorganic nitrogen source. Organic nitrogen source is a yeast, peptone, and tryptone or the other the protein degrading the compound. And phosphorus and the mineral also important, but not so important like the carbon and the nitrogen source. So phosphorus is the crucial for energy transfer and the nucleic acid DNA synthesis. Other minerals are also essential for growth and metabolism. And the vitamin and the growth factor also important for the growth of the transcriptase because of the optimal growth and the lipid production. So next is what kind of the environment factor and the stress influence to the transcriptase. First one is the temperature. Trust criteria is very sensitive to the temperature changes. Optimal the growth the occur in moderate temperature like the 25 to 35 like this or the 20 also can acceptable. But normally the my lab case is the I set at 28 degrees Celsius because this one is just we follow the environmental the temperature. But they can tolerate the range of the condition. So that's why the some the trust criteria can isolate from the Antarctic environment. And salinity, trust criteria are tolerant microorganisms. That means that they can survive in the variable the salinities. And this salinity also influences their growth, lipid accumulation, and the production of the bioactive the compound. So the normally the researcher check the how much the 30 ppt is the normally the marine environmental salinity. So 30 ppt or 40 ppt or 20 ppt or 10 ppt. How the this kind of variety of the salinity influence their growth and the lipid and the other bioactive compound. And nutrient ability is availability, I already told you. And the oxygen also important because the trust are generally aerobic. That means that they need oxygen for growth. But they can tolerate low oxygen level. But the prolonged hypoxia can negatively impact their survival. So what is the challenge and the future direction? So genomic and the metabolomics need for the how their metabolic pathway and how their genetic diversity. And the metabolic engineering also important to the develop the advance in the metabolic engineering can optimize the lipid and the other bioactive compound harvesting the products. And the cultivation optimization also important, such the fermentation or the bioreactor design is the vital for scaling up for transcriptive production like the lipid or enzyme or bioactive compound. And the regulation and the safety, of course, if they want to make the products, of course, we need to consider the regulation and the safety. So far, the known of the transcriptive is a pathogen, and their products should be safety, but we need to consider this kind of the regulation and the safety for the commercialize. So the wider grow commercialize and the market trends is the growing the demand because as I said, they produce produce a lot of the PUFA, like the DHA, EPA. Nowadays, the aquaculture diet, we use the fish oil. So fish, because the fish oil compound a lot of the PUFA. But nowadays are increasing the fish oil price. So if we can replace the transcyclic oil with the fish oil, then we easily to get and the cheap price of the aquaculture. Uh, Also the food or the pharmaceutical and the biofresh also. So until now is the like the general information about what is the transcriptase and why I do the transcriptase. So next, from the next slide is the actual my experiment. Okay. So at first isolation and the characterization of transcriptase from marine water. In this study, so the following this study is the all how many that one, two, five UNDIP students involved in this the research. So first student conduct the isolation. One student conduct the isolation and the characterization of the transcriptase from marine waters. So as you can see that this one, upper one is the transcriptase. transcritic colony using the microscope. And the bottom one is the transcritic cell using the microscope. So, as you can see, the easy to see that their cell, even though the colony. Then this is the sequence result and their how to say that pure lipid compound. So as you can see, the three isolates is a compose the okay, same cluster with the oblongichytrium species. Okay, and even though the same cluster, the same species, their SFA, MIFA, PIFA compounds are totally different. For the example, the MIFA dominant, the largest category, the more than 50% of the lipid composed of the MIFA. And the PIFA is the second highest, around 30 to 30, 27 to 20, 38, 5%. And the sample S41, so the S41, you can see the sample size for the having the most fatty PFA, PFA composed of the EPA is highest, and the ALA, alpha linoleic acid, also highest compound in the S41, and the S62 is the oleic acid. Only S62 produce the oleic acid. And T62 is the highest SFA compound producer. And they produce a stearic acid, but they don't produce the palmitic acid. Okay. How to say that? Even though the same species, but a different strain show the different lipid compound. So next is the isolation and the characterization of the transcriptase from the mangrove leaf and the sediment. This one is the three UNDIP students involved this research. So this one is we isolated 9 transcriptase. And you can see this one, 1, 2, 3, 4, 5, 6 strain is the belong to the Schizochytrium species. And 2 isolates is similar to the Transcytiridae. And 1 isolate is a Botryocytrium. And this S is a sediment. And the leaf, L is a leaf. So as you can see, only the sediment identify the Botryocytrium. But the leaf sample is a 2 different genus. So let's see how their lipid compound. So at first, the overall, the profile is extremely top-heavy flavored saturated fatty acid. This one is more than almost 80%, and some days 95% is composed of the saturated acid. Already different with the oblongichytrium. Oblongichytrium is around 50% is the MUFA and the 30% is the PUFA. Then saturated fatty acid the lowest. But in this study is the totally different. So saturated fatty acid the make up the vast majority of the in all the sample like the 73% until the 96%. And the dominant of the saturated fat acid. ラベル。here か。palmitic acid and the stearic acid is the dominant of the, no, sorry, dominant is the palmitic acid of the all of the eight isolates. So this one. And the palmitoic acid, the C16 is the most highly priced fat for standard biodiesel. And the palmitic acid and the stearic acid, this, these two is a very high production in the all the isolate. Is the, how to say that, is the incredibly well and the high energy density. And others is the C16, yeah, palmitic acid, yeah. Okay, go. And the palmitic acid and the stearic acid are used as a phase change materials in green building construction, and that one is, the absorb the heat during the daytime. And next is the, which one? This one. Oh, next is the linoleic acid. Also, in the prefer also the very different, even though the same species, sorry, forget to tell. L311 and L332 is the same until here. L141, sorry, 411 is the same the cytokine, but the total different among them of the lipid compound. Okay. Then linoleic acid is okay. Linoleic acid here. Linoleic acid and which one? The arachidonic acid. Yeah, okay, okay. GLEP, no. Linoleic acid, arachidonic acid. This one. Sorry, I forget to the mark in here. The one of the potential diesel oil, biodiesel. And other than the biodiesel, which one? Here the ALA, and the EPA or the GLA are the extracted purify and sold at a massive premium to the pharmaceutical and the nutraceutical industries. And also oleic acid and the arachidonic acid is a very important for the, how to say that, supplement. So that as you can see, the even same species, but a different compound. So that's why when we want to commercialize or develop the products, the strain selection is a very important. Example that if we want to target palmitic acid, then if we choose this 312, this is the lowest. So that means the final products is the lowest final products. So if we want to focus on the palmitic acid, need to choose the L332 because they produce the highest the palmitic acid compound. And if want to focus on the EPA, then we cannot choose the 332 because the lowest. So we need to choose the 312. That's why the strain strain selection is very important for the commercialize. And next is the how the importance of the nutrient source. In this study, we focus only the nitrogen, organic nitrogen compound. So P is a protein, peptone. T is a tryptophan, and Y is a yeast. So as you can see, the tryptophan show the highest saturated fatty acid, and the caprylic acid also highest, and the lauric acid also lauric acid highest in the peptone. And you can see the mu5 is the lowest. And the yeast is the highest production of the PUFA, like the arachidonic acid or the EPA is the highest in the yeast. So this case is that if we want, that's why if we want to focus the EPA, maybe we need to use the yeast or the tryptophan or peptone, but this one is depend on the strain. So once that we choose the strain, then we need to check the nutrition. Nitrogen source, which nitrogen source is the highest production in the EPA or MUFA or SFA. かな ?OK かな、うん、So the PAP is the sample P peptone stands out for having the highest concentration of the lauric acid. This one is the one of the biosurfactant. And they show the antimicrobial effect. And sample, sorry, the tryptophan treatment, high unique for immersive in the caprylic acid C80 is a more the double of the others, and also the lowest total MUFA. And each treatment is the most unsaturated sample of the three, so the unsaturated so highest in the PUFA and the MUFA, and the highest in the DGLA, this one, GLA. Sorry, DGLA is this one, sorry, 17.84%, but almost similar with the others. So this one is how important for the selection, strain selection, and nutrition selection. Then next is I use the Traskytri as the bioelimination. So at first that we want to use this one. Sorry, no yeah. Sorry. Then next one is the isolate from the aquaculture sludge. So this one is a one undip student involved. So that the student in isolate the three isolate, and all three is a Traskytrium species. And their how to say that? The lipid compound also a bit different, but almost similar, these of three. But the inside, like the SFA is almost a 60%, and MUFA is a 20, and Q5 is a 10 to 15%, 70%. So the not so drastically different compared to the previous isolates. However, the palmitic acid or the palmitoleic acid or the oleic acid and the ALA or the EPA compounds are totally different among the strains. So palmitic acid is the highest in the SH 1.2, and the palmitoleic acid is the highest in the SH 4.2, and the oleic acid is the ALA, alpha linoleic acid, and the EPA is the highest in the K1. Okay, so until now is just I share about isolation and the nutritional selection. Next is we use the isolated transcriptase as the bioelimination, because as you all know, nowadays SDG. So we cannot, no, not only we, the industry or the local shop also cannot easily to throw their wastewater because the wastewater compound a lot of the nutrient, high carbon source and the nitrogen source. So if we directly throw to the environment, maybe the later on the pollutant. So, and the trash treat consider can use these kind of high nutrient. So we want to try whether the trash treat can use the aquaculture wastewater as their growth source, growth media. So this is the result. So green one is a control. That means just the normal media. And red and blue is the wastewater, but a different wastewater source. So as you can see, even though the lower growth compared to the control, the media, trash can grow using the wastewater. And why this one, you can see why the boil and the wash is low compared to the glucose, gross media, might be the salinity and pH. As I say their optimum pH is around 6.5 to 7.5, and salinity also important. So if the zero PPT is a bit difficult for them to grow, that's why maybe their growth is suppressed by either the pH or salinity. Then next is this one is next is the lipid compound, especially the PFA compound. So the growth media, just the media, is a high composition of the DHA. However, the wash, how to say this one, linoleic acid and the linolenic acid and the eicosanoic acid production is high in wash sample. And EPA is the only product from the boil sample. So that means this kind of stress, environmental stress, when the transcriptors feel the stress, maybe they produce a different type of the variety type of the lipid, not only the DHA. So conclusively, we can say the transcriptors can grow using the wastewater. And next is this one is aquaculture wastewater. So control is the normal media. Treatment one is the 50% aquaculture wastewater, 50% media, and 100 is 100% aquaculture waste. And you can see that almost pH is similar. However, this one, you can see the 100% aquaculture waste is lower compared to the 50-50 or control. That might be the aquaculture waste nutrient is the low or too high. That's why we need to check what kind of nutrient source, nutrient compound in the aquaculture waste. So if we know what kind of compound, nitrogen or phosphorus, or that they are enough or excess, then we adjust, maybe we can use the aquaculture wastewater as well. So this is the lipid compound from each treatment. So this one DHA compound is a lower or almost zero in the 100% aquaculture waste. However, the 50-50 is an increase to the DHA production. So we can use the trans as the aquaculture waste or the food processing wastewater as the bioelimination or biodegradation the organisms. Okay. Then last one is the we also conduct next generation sequence, not only isolation, unculturable the transcriptory compound from the coastal area. So this is the coastal area water result. As you can see, a bit small ka. A bit small, but so you can see that if the near the river mouth and far from the river mouth, this one is around 50 kilometer, 50 kilometer far from the coastal area to the river mouth. So you can see the one, two. So these two and these three bacteria, transcriptory conversions are totally different. So this one can see the as the same things. So you can see the station 345 345 is higher in the Aplanocytorium. However, the station 1 and 2, this one 1 and 2, is the low in Aplanocytorium, but the N, you can see the unclassified or uncultured Trastocytoridae. So that means the coastal water is still a lot of the unknown Trastocytrid exists in the coastal water. So in the conclusion of the Trastocytrid community, predominant is the Parancytridium and unclassified Trastocytrid cell. And their diversity is a station 1 and 2 is far from the river mouth, is distinct bacterial diversity, Trastocytrid diversity with the other station. That's all my today's the presentation. Okay. Okay. Miss Arifa? え、コネクトか。今 Miss Arifa?
Okay, thank you so much, Dr. Sumpe, for the insightful presentation. So we are going to the Q&A session. For student who has the question for Dr. Sumpe, please give the question. Is there any student who have question for Dr. Sumpe? Okay, Nova.
Thank you so much, Ualifa, for this piece. And let me ask Mr. Sumpe question about this topic. So before applying Taus-Citrus on a large scale for bioremediation, it is important to think not only about the benefit, but also about the possible effect on natural water ecosystem. So is there any ethical or ecological risks that may arise from introducing large population of Taus-Citrus into natural waters? Is that clear?
Sorry, I cannot hear properly, clearly. Can you give that again?
Okay, so is there any ethical or ecological risks that may arise from introducing large population of Taus-citrus into natural waters?
Ethical and
Ethical or ecological risks?
ecological ethics.
Ecological.
Ecological ethics.
Ecological risks. Resiko in ekologi.
Risk. Ecological risk.
Ya.
Teruskan population on the bioremediation. Ecological risk. That one is I'm not sure because still the many researcher the start how to say that start using the Truska treat or the still they don't use yet. I also not yet. So my case is that I just use the aquaculture wastewater and seafood safety, seafood processing water in the lab scale. So of course the maybe the bioelimination is already developed in the bacteria researcher, but of course the small scale, the large scale is a different. So the large scale in the with mean the natural condition is a different. Lab scale and the natural scale is a different. So maybe have the some the risk like the if the put the trans then maybe change the natural microbial composition. But this one also the still the need to do the investigate the more longer, much detail. Nafa, is it answer for your question?
Yes, thank you so much, Deskina.
Okay. Okay, thank you, Naufal, and also Dr. Sumpe. Any question for the students or the participant in the UGC session today? We still have time to the Q&A session. If students have question for Dr. Sumpe, please give the question.
So that this transcriptory research is still the latest one, not like the bacteria or other
Mm.
microorganisms. So the many things we need to do. So we cannot say the imply to the natural condition also, so that we also know the natural what kind of risk, environment risk or something. But we cannot say so far. And nowadays we still focusing on the isolation and optimization of the Terrasky tree and using the NGS also. So because we don't know yet what kind of Terrasky tree actually inhibit in the environmental sample. Because normally that we do the many researcher focus on the mangrove reef, not only not focusing on the coastal water.
Mm. Okay, Dr. Sumpe. But I'm curious about the research of the toxicity because many researchers get the toxicity from the mangrove area. Is it possible to get the toxicity in the coastal area or maybe in the lake?
Coastal area can,
Okay.
yeah, can, but as I showed, the Planocytorium is
Hmm.
the how to say that plankton eating. Their prey is the plankton. So if
Oh.
you want to isolate of the Planocytorium or something, then you need to optimize the isolation method also. So maybe we cannot use the isolation method to the with the mangrove reef one. Maybe need to change the method
Hmm.
because they are not totally some of the Truskatri live in the coastal water, but maybe the totally different. So we need to optimize because actually my student try to isolate the Truskatri from the coastal water, but cannot isolate. Maybe the 4 or 5 times go to the sampling. Then try to isolate but cannot.
Hmm.
Yeah, but only we can isolate from the beach area, not the coastal, very near to the us. it can isolate.
Okay.
That's why I want to know what kind of trust inhabit in the coastal area. So the last slide one. So that if the coastal area one is the unknown trust. So unknown means that we don't know how to isolate.
Oh,
And
yeah.
many bacteria also same. So that I do the coral reef one also. So that many the bacteria inside the marine sponge or coral cannot culture. We know that this bacteria inside the marine sponge or coral area, but we cannot isolate because we don't know what kind of their diet, food.
Mm.
So same thing can say to the Trascatrid. So maybe in future we need to do that.
Yeah, yeah. Maybe we can do it. Yeah.
Yeah.
I'm curious about the categories of the Trousocetid. Is it yeast or the or diatom or algae?
No,
No,
is
not
no,
of all.
not at all. Because if the is a very small, but the
Mm.
size is a very bigger in the Trascatrid. And diatom, they have the silica. The shell
Mm.
is a silica. But the Trascatrid is like the chitin. And the microalgae, they do the photosynthesis. Trascatrid, not photosynthesis.
Ah.
That's why Trascatrid is a fungi-like protist.
So Trousocetid fungi but like protists.
Fungi-like. Yeah,
Okay.
because they are fungi and Trascatrid is a decomposer. And
Okay.
colony also looks like the fungi. So fungi-like protist, or just we say the Trascatrid or Labyrintula.
Okay. 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 Okay, for student, is there any question for Dr. Sumpei? We still have time. If you have any question, please raise your hand for Dr. Sumpei, or you can also chat in the into the chat box. Okay, okay, Dr. Sumpei. May I know when you first researched about the Tau society, why you finally want to know about the Tau society?
Because actually my senior in Japan do the Trascatrid. Then I talk with him. Then I search the journal. Then I noticed that nobody was only few researcher do in the Malaysia and Indonesia or the ASEAN country. Mainly the Thai, India, or Japan or USA researcher only do, or the Australian country. So not many the ASEAN country researcher conduct the transcriptid. And transcriptid have a lot of the potential, like the PUFA or the carotenoid or the enzyme. So my originality is a probiotics. So, but
Oh,
transcriptid also can consider probiotics because the probiotics is the improve the immune system or the improve the growth or the improve the nutrition in the host. So Trascatri also similar. If we give the Trascatri, then if the host growth then improve or the immune is improved. So for me it's a similar. So I want to focus on the Trascatri. Of course, not only the Trascatri, I also conduct the microbiome, like the last week that I in my presentation. So far I do the both microorganism, microbiome, and the Trascatri.
oh yeah, yeah. It's so interesting because nowadays I research about the lactic acid bacteria also from
Mhm.
the fermentation food, and trousocited I think has the potential for the PUFA also. So we can consume the trousocitrate like a supplement or something like that.
If the not transcribe themselves as a supplement, if the human,
Oh,
maybe we can give to the fish diet. Of course, if the we want to give the
yeah.
produce the transcriptory supplement as a human supplement, maybe that we need to the safety or regulation. But fish or something is still low, not so many regulation, and because just that we see the whether the safety or not only. Of course, the human is a more high regulation.
Okay.
So maybe that if they want to use the human supplement as a human, maybe the just transcribe not transcribe themselves. Maybe transcribe products like the PFACA or squalenca carotenoid. Just the extracts, not use the products. Sorry, the transcribe themselves.
Okay. 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 There is a question from Aulia Zalianti. I will read the question from Aulia Zalianti for Dr. Sumpei. Okay, this is the question is about the remediation with polarization. How does the concept of remediation with polarization change the economics of pollution management?
As you know, the bioremediation is the how to say clean up the pollutant using the microorganisms. So bioremediation is the very good because if we use the mechanical or something, then sometimes the how to say mechanical one is take the cost, but if the bioremediation using the microbes is the low cost and not so the drastically changes their environment, natural environment is okay. So that not so familiar to that side also yet.
Okay. Yeah, okay. Okay, thank you for the answering, Mr. Sumpei. And now for the student, is there any question? Okay. If there is no question, so we'll move to the final session today because this is the last session of the UGC with Dr. Sumpei. Before we close the UGC today, maybe from Dr. Sumpei, there is any suggestion for the students?
Okay, but before the suggestion, I want to say the appreciation to the five UNDIP students, those who study, conduct FYP under me. Without their support or their research, maybe I cannot make that today's slide research and maybe the future in the publication. So that I really appreciate to the student, UNDIP student to help my research. And so if you interest into the transcriptory research or the last week the microbiome research, gut microbiome or the water microbiome, just contact Pak Anto. Yeah, I accept anytime.
Okay, it's so interesting informations. Yeah, I know one of the student that go to the UMT,
Mhm.
Monica, right?
Yeah, Monique is now Monique is just come.
Yeah, okay. I
Yeah.
know Monica so well. So yeah, thank you
Uh-huh.
so much, Dr. Sumpei, for your insightful suggestion. So for our students who want to research about the thrombosis rate or all the research that Dr. Sumpei can do, you can contact Pak Anto. Maybe for the scriptsy or also the thesis one, if you want to do the research with Dr. Sumpei. Okay. This is the last session of UGC today. I would like to express our deepest gratitude to the Dr. Sumpei Ihata for UMT for the insightful, also the interesting explanation about the microbial, also the Trau Society today. Also, I would like to express thank you very much for the all participant today. Thank you also to the lecturer in the microbiology industry, Professor Dr. Endang Kusdiantini, and also Ibu Dr. Arina Trilunggani. So we hope this session has inspired you and will open doors to the future collaboration for UMT and also for Diponegoro University, because nowadays there are 4 students for UMT in the Diponegoro University also. This is the last session of our UGC. Thank you so much. Assalamualaikum warahmatullahi wabarakatuh.
Thank you. Thank you
Thank
very
you
much.
so much, Dr. Sumpe.
Thank you. See you.